Jannavi. Here at Evolutionary Ecology Lab, we study biodiversity around us. We ask questions like how many species are there, how have species evolved, why species are where they are, how ecology, geology, climate has shaped their evolutionary history. And to address these questions, we use multiple model systems, including terrestrial soil arthropods, woody plants, birds in the Indian tropical forest, which are exceptionally biodiverse.
Systematics is the study of classifying different kinds of organisms with an evolutionary perspective, and the lowest unit of classification is called a species. During the process of speciation, a new species develops changes across various lines of evidences, and most of which is morphological, which is why most of taxonomic knowledge is based on morphological differences. But there are other lines to it as well, be it on the lines of genetic differences or the chemical composition of the species. In the lab, we use multiple lines of evidences to describe and identify different species.
So one of the key aims of the lab is to look at the origin and distribution of lineages over evolutionary time period, and this is termed as historical biogeography. And we use Peninsular India as a case study to examine how deep past processes have shaped the current diversity and distribution of lineages. And we do this by combining information on the molecular data, fossil, and also current distribution to examine how these past processes have shaped where organisms are currently distributed and how they have diversified.
Furthermore, we intend to understand how organisms have speciated, gone extinct, and have assembled within a certain landscape. Peninsular Indian landscape is the model system in our lab, and we essentially look at organisms that exist in the Western Ghats biodiversity hotspot. We use time-calibrated phylogenies, which have been calibrated using fossils and using molecular data from the DNA. And using these time-calibrated phylogenies, we understand how organisms have increased in the number of species over time, and how have they assembled across environmental climatic gradient within the Western Ghats and across the peninsular India.
So apart from studying all these biogeography and species diversification studies in our lab, what we also try to understand is how traits evolve in nature and what are the ecological as well as evolutionary drivers that govern these trait dynamics. And one such excellent model system which is very interesting to work with is animal venoms. And in our lab, we work with centipede venoms, trying to understand how they have evolved and what are the potential drivers that are governing their dynamics. So basically, when any live animal comes to the lab, we first extract their venoms by giving electric current, and the entire process is mediated by something which is known as a TENS unit, also known as trans-electrical neurostimulation unit. After the venoms are extracted, we then do their dissections and do their venom gland transcriptomics, integrate the transcriptome and proteome study to understand how these traits evolve. So once their venoms are extracted and we have dissected their tissues, we use sophisticated molecular tools such as mass spectrometry and gland transcriptomics to understand that as a whole how centipede venoms have evolved, to document their diversity across multiple locations in peninsular India. So apart from this, we are also trying to understand that how diet influences venom evolution and how is venom complexity related or correlated with the diet complexity. And for that, we also use sophisticated tools such as gut metabarcoding, again with these centipedes as our model system.
In a nutshell, we aim to understand how biodiversity has originated and is being maintained over millions of years and at a large spatial scales like mountain range or a continent.